In our discussions on stem cells and senescence, we already mentioned telomeres. So now it's time to actually talk about the telomeres themselves. In contrast to prokaryotes, who have a circular chromosome, eukaryotic cells have linear chromosomes. The end structure of these linear chromosomes are called telomeres. On the level of DNA itself, telomeres contain highly repetitive DNA sequences that are usually very CG-rich. The precise sequence of these so-called tandem repeats differs from species to species, but the consensus sequence is TTAGGG. This short DNA sequence is repeated hundreds to thousands of times, so that the total length of the telomeric DNA sequences is often several thousand kilobases long. In humans, for example, telomere sequences at birth are around 10 kilobases long, while mice have a telomere length between 30 to 50 kilobases. One important feature of the telomere DNA is the presence of a 3' overhang, which means that at the chromosome ends, DNA exists only in a single-stranded form, instead of the familiar Watson-Crick type DNA double helix. In order to protect this 3' overhang from degradation, which is what usually happens to single-stranded DNA inside a cell, the overhang is able to loop back and invade regular telomeric double-stranded DNA upstream in the telomere sequence to form a structure termed the T-loop. The formation of this T-loop structure is critically dependent on a multi-protein complex called shelterin. Shelterin consists of a variety of proteins which recognize specific DNA sequences and binds and stabilizes the various parts of the telomeres, from the single-stranded overhangs to the double-stranded and invaded parts. Thus, the proteins of the shelterin complex provide a form of cap on the telomere ends that protects its telomere from degradation by cellular enzymes. To a cell, an uncapped or non-T-loop telomere looks exactly like damaged DNA. It is a single-stranded piece of DNA instead of double-stranded, similar to how DNA breaks appear. DNA damage can be very efficiently repaired by a cell via a variety of mechanisms that may include the degradation, or sometimes also referred to as erosion, of the single-stranded DNA, as well as some adjacent double-stranded portions. This in turn could lead to telomere shrinkage. Alternatively, some DNA repair processes simply fuse a piece of double-stranded DNA to another in order to repair the damaged chromosomes. This could potentially result in individual chromosomes being fused together. The end result in either of these scenarios would be a drastically altered chromosome and telomere structure, which could have disastrous consequences for the cell. During the S phase of the cell cycle, DNA gets copied or replicated. Chemically, this process must always occur by adding new nucleotide on the 3' end of the growing daughter strand. When there are no 3' nucleotides present, a specialized enzyme creates an RNA primer to serve as an attachment point for the new nucleotides to be added. This primer is subsequently degraded and replaced by DNA. At the end of the chromosome, this primer degradation creates a gap that simply cannot be filled anymore. In other words, the chromosomal ends cannot be completely replicated and thus shrink with each cell division. If the telomeres shrink below a certain size, they are simply no longer able to form T-loops and remain open for degradation. The whole chromosome thus becomes unstable, replicates poorly, if at all, and the cell may consequently die. This is the basis for the Hayflick limit that we discussed in an earlier module, which states that cells have limited replicative potential, maybe around 50 or so cell divisions, depending on the particular cell type. To work around this so-called end replication problem, eukaryotic cells have a specialized enzyme called telomerase, or TERT. Telomerase is a very special enzyme in that it contains an RNA component called TERC, or TERC. This RNA serves as a template for TERT to add new nucleotides to the ends of chromosomes. In this way, telomerase adds new nucleotides to the 3' overhang of the telomere sequence, while the complementary strand then is filled in by the regular DNA polymerase enzymes using the newly elongated 3' overhang strand as a template. Thus, telomerase does not really prevent shrinkage of telomeres per se, as that can't be avoided due to the chemistry of the replication process. Instead, it can grow back the telomere length to its cell-specific size. Cells that have active telomerase can therefore evade the Hayflick limit. Most cancers are, in fact, telomerase positive and are essentially immortalized. Most somatic cells are telomerase negative in order for the cells not to overproliferate within their organ and tissue context. Cells that depend heavily on their proliferative capacity, such as stem cells or certain cells of the immune system, have elevated telomerase activity. As described earlier, when the telomere is damaged or critically shortened, the cell is no longer able to distinguish a telomere structure from simply damaged DNA, and will thus try to repair the damage using the standard DNA repair mechanisms. What works for normal DNA damage, however, is unfortunately exacerbating the problem at telomeres, as it may, as noted earlier, lead to further shrinkage or even chromosomal fusions. When fusion occurs, chromosomes may break into even smaller pieces during mitosis, as these di- or even polycentric chromosomes will be incorrectly attached to the mitotic spindle machinery, which in turn leads to further chromosomal and genetic instability, and subsequently loss of this genetic material. In any case, the damage remains in one form or another. And now, the DNA damage checkpoint proteins that we have met earlier in the chapter on cancer become activated to determine the ultimate cell fate. If repair of the DNA lesion can be initiated and executed in an error-free way, no further problems exist and the cell remains healthy. In the case of damaged telomeres, that repair often is faulty, as we talked about earlier, and may result in a cancerous cell. The paradox that exists here is that they are rapidly dividing and dividing indefinitely. If telomere shrinkage leads to chromosomal instability and cell death, how can cancer cells overcome this instability? The answer is that cancer cells often have disabled DNA damage checkpoint proteins, such as p53, that enable them to escape from the induction of apoptotic or senescence mechanisms. Still, during each cell division, cancer cell chromosomes should shrink further until they are no longer functional. Cancer cell may evade that fate by either having additional mutations that, for example, could activate TERT, or by an alternative telomere lengthening system termed ALT, for alternative lengthening of telomeres. This ALT uses the process of homologous recombination to essentially copy intact telomeres from one chromosome to another. If repair cannot be performed, the checkpoint proteins, especially the p53 pathway, which we discussed earlier, may then opt for two ways to remove such a damaged cell to eliminate it as a threat to the organism. This occurs either by inducing the cell to commit suicide or to isolate the cell via induction of cellular senescence. Once again, this is a two-edged sword, unfortunately. While it maintains, at least for now, organismal health by removing the damaged cells, it may also contribute to aging phenotypes by, ironically enough, removing cells from a tissue that may be needed for proper tissue function. With telomeres playing such an important role in the balancing of genomic stability, cancer development, and tissue function, it is no surprise then that they also may play an important role in the aging process. If that reasoning is correct, the animals deficient in TERT, the enzyme responsible for telomere maintenance, should have lower replicative potential, increased tissue aging, and decreased longevity. The initial experiments in mice in this regard were very disappointing, unfortunately. Telomerase deficient mice showed no obvious phenotypes. This, however, may be due to the exceptional length of telomeres in mice, which are around 4 times longer than human telomeres. When TERT knockout mice are bred for several generations, researchers finally observed telomere shrinkage, along with stem cell dysfunction, generally accelerated aging phenotypes, and shortened longevity, pretty much as expected. The key experiment then was to reactivate telomerase in these prematurely aging animals. And surely enough, this intervention completely reversed the accelerated aging phenotypes, providing direct evidence that telomere dysfunction does indeed contribute to aging. Telomere length per se, however, does not give an indication of longevity. Remember, mouse telomeres are much longer than human telomeres, yet mice have much shorter lifespans than humans. But within a species, telomere length could be used to determine tissue aging and health. In this context, longer telomeres are usually associated with better general health, while shorter telomeres are often found in damaged tissues. While the mechanisms are still unclear, it is apparent that a lot of external stressors do in fact result in shortened telomeres, such as smoking, poor nutrition, or xenobiotics. Possibly, this may occur via the generation of reactive oxygen species. Altogether, this makes telomere extension an attractive approach in possibly arresting age-related pathologies. Nonetheless, it has so far not been shown that simply overactivating telomerase may extend longevity, which may be problematic regardless, as higher telomerase activity does in fact lead to increased cancer incidence. Once again, the two-edged sword. Check your knowledge. Describe telomere structure and function. Explain telomerase function. Describe how telomeres shrink. Explain the consequences of telomere dysfunction. Explain how telomeres are connected to cancer and aging.